Hi everyone, thank you for the people that are not on the beach now, the few of us. I've been in an argument in my faculty months ago if it's closing remarks or just the last one that nobody wants really to hear. So I will call it closing remarks for my own, self-esteem. So thank you very much to the people that stay with me here. Let me see how this works. Like that. Okay. So my name is Dedi Meiri. I'm a professor at the Technion, the Technology Institute of Israel. As a disclaimer, also owner of Cannasol, which is a company that's doing analytics, but also a lot in the R&D, and part of the results that I will show you here have been done also in Cannasol. Cannabis. I think it's stupid to show this figure here in the last session of the day, and also to say that is a unique plant that harboring unique molecule called cannabinoids, and it's affecting the endocannabinoid system in our body. But I'm saying that to people that maybe not admire how complicated the endocannabinoid system or how complicated cannabis is. The endocannabinoid system is a system in our body that every cell in our body harboring the endocannabinoid system and being affected by endocannabinoids. So there is 220 some different types of cells in our body, whether these are neuron, whether these are skin cells or liver cells, they're all harboring receptors for the endocannabinoid system and can produce endocannabinoids. In the blood samples today, we can analyze more than 120 different ligand endocannabinoids, and there are more than 30 receptors that we are looking on them. Actually, the endocannabinoid system, it's a communication between two cells. One cell sends a signal to another cell, increase appetite, reduce pain, increase heart beating, modulation of glucose modulation or glucose intake, immune cells. They're all being controlled by the endocannabinoid system. We didn't say any word till now about cannabis. It's a system in our body that control and balance, doing kind of a homeostasis on almost all the cells in our body. Cannabis was just totally unlucky to produce similar molecules to the molecules that our body producing. More than 10,000 years ago, people started to breed this plant and to increase the amount of these molecules and create kind of a puppy, kind of a poodle plant. It's not a natural plant, okay? I know that there is people from Morocco here that told me you need to come and to see the original plant. Guys, 10,000 years ago, people walk with that, breed it. There is no original. It's all been produced by humans. The cannabis shouldn't produce more than 0.1, 0.2% of phytocannabinoids. Lumir Hanush, who is here, published a few years ago about 60 some plants that producing phytocannabinoids. The difference between them to cannabis is the amount of phytocannabinoids. Cannabis was breeded and breeded and breeded, and we create a plant that harbor a lot of these molecules in high percentage. People from the 60s can tell you that in Woodstock it was 6% THC and not 36% THC. Why? We didn't do the breeding right, that's all, okay? So, cannabis, it's not a one plant. It's a lot of, I will call them chemovars, different variation that harboring different combination of these molecules. And in my lab, we identified 11 different families, and we can analyze around 166 phytocannabinoids. And the big question in this arena, and this is similar to what I talked last year, but I will take it from a different angle soon. The big question is how you match the right flower to the right indication, how you match the right flower to the right bride, right? If cannabis or the endocannabinoids can influence pain, sleep, appetite, immune, glucose, So which combination of molecule, which plant can affect the appetite, which one affecting immune, or which one of them reducing pain. So how we are doing that, and I show these results enough time in CannMed, so I would go fast to reach the point that I want to talk about it. So how we can do that? We can screen on cell culture, we can take data from patient, we can do many things to define which cannabis is affecting which indication. So if we're screening, we can grow cells on the plate like that. We can grow cells on the plate that have many wells. We can have equipment that's screening it very fast. In the lower plate, you can see 96 well plate that have 96 well. You can put different types of cannabis, different dosing, and there is equipment that's screening very fast. I won't go on that. It's not important. And these are results from my lab. Most of you already saw that result. I'm showing that in the last 3 years, so I'm going fast on them. This is how cannabis influences leukemia. We know that cannabis affecting the immune cells. So how it's affecting the cancer of the immune cells. So Eli Besser, PhD in my lab, take a TLL, which is a specific type of leukemia, screen of them, different chemovar of cannabis. You see every color, it's a different chemovar. And you see when the line is lower, that means that it kills more cells. So you see that in 2 micrograms per ml, most of the cannabis didn't affect the leukemia, but there was one extract that was very efficient killing the leukemia. Because leukemia, it's a given name to many different diseases. We tested different types of leukemias, and we saw that in 50% of them, these types of cannabis kill the leukemia very efficiently. In 50% of them, it didn't affect. Actually, the opposite, what I showed, 50% didn't affect, and 50% it killed very efficiently. So we screened this 10 leukemia, we did a genomic screening, and we find that every time that there is a specific mutation called Notch1, this specific cannabis killing the cells. When every time that there is no mutation, it's not affecting the cells. What I'm trying to show you is how specific it is. Cannabis can kill cancer, Rick Simpson oil? No. One type of cannabis from 200 and some different chemovars that we test was the only one that affects this leukemia. From that, it's not all the leukemias, it's just leukemia that harboring specific mutation. And this Notch1 is a protein sitting on the membrane, feel the other cells near him, what we call contact inhibition. Doesn't important. We take this specific leukemia, we create mice model for leukemia. You can see a CT of mice with a cancer in his back. We're treating these mice with cannabis, with this specific extract. You can see the black line on the left. It's tumors with mice that didn't treat with cannabis, how the tumor are growing exponentially, while the gray line is mice that harbor tumors that treated with this specific cannabis, which the tumors didn't grow. On the right side, you can see the tumors before and after 3 weeks treatment. Because this is leukemia, we did the bone marrow implantation with primary cells from patient. We show that we are doubling the lifespan of the mice. We did 5 different mice models, and we show that. Now we're asking which molecules from the cannabis doing this effect. These are all the molecules that this specific extract is harboring. We're taking this extract, we fractionate it, we're taking pieces. We said there is 100 molecules that take 25, 25, 25, and 25. We're going back to the cells and the mice, and you see that the first line, the vehicle, didn't kill the cells. The whole extract number 10 was very efficient, killing the cells, and then 4 fraction, and just fraction number 2 mimic the whole extract. So we're already eliminating 75% of the molecules that we don't need. And we're going back and forth, and what's important here is that in the end we succeed to analyze 3 molecules, just 3, that if you take them synthetic or purified from the plant and take the 3 of them, you get exactly the same action as the whole extract. If you take any one of them, and this is the bars in the left low, every one of them, or combination of 2 of them, it's not working as good as the 3 of them together, okay? And we did a lot of cell biology and biochemistry, and this paper actually published last week. We did a lot of work. In the end, we define exactly the mechanism of action. These 3 molecules, CBD, CBDV, and a new cannabinoids, we call it 3318, binding two different receptors, CB2 and TRP1, and doing a full action of many proteins, and I won't get into that now, that actually blocking in the Golgi the ability of the mutant protein to reach to the membrane. A lot of biology, when most of you are not scientists and not physicians, which is not important to you. What I want to say, we understand why we need three molecules, and we understand what they are doing the cells, and why there is mutation, it's killing the cells, and when there is no mutation, it's not killing the cells. We bridging the gap between herbal medicine, between saying this is a plant that's affecting, to real modern science and modern medicine showing these are the molecules, this is the affinity, this is the avidity, this is what they are doing, this is why you need the three of them. But pay attention, we need three of them. Most of the drugs that have been approved by the FDA, it's single molecule to single target. Why? Because we're afraid from drug-drug interaction and more things. This is kind of next level. I can show with different work that sometimes we can isolate single molecules from cannabis. In that slide we see from breast cancer, different types of cancer, we isolate different type of molecule, we call it 37315B. It's similar a little bit to CBG, but you can say that there is 3 oxygen there, and this molecule reduces the activity of the estrogen receptor, and for that reason, in estrogen receptor, in breast cancer, dependent estrogen receptors, it can affect the cells, okay? Again, not important the breast cancer and the results, the idea. In this case, it's one molecule. It's not THC, not CBD, not CBG. You can call it minor cannabinoids. It won't be minor if you're a breast cancer patient, okay? It's a molecule that reduces the estrogen, and for that it can affect estrogen. But this is now, it's one molecule. So I showed you that cannabis can affect cancer in different ways, different cancer, different cannabis. Is it always good? So this is a different question on drug-drug interaction. What are these molecules? We know nothing about this molecule. We don't know if it's safe, if it's not safe. But we asked a few years ago, we know that cannabis modulates the immune system. We know that it's good for inflammatory bowel disease. We know that for autoimmune disease, patients in Israel being treated with cannabis. And I showed you that it's affecting T cells in leukemia. One of the best treatment today in cancer, or the most innovative treatment, is in immunotherapy. Immunotherapy is when we are telling or teaching the immune cells to attack the cancer cells. The cancer doing two things, and I will show a slide in a moment. He's changing the microenvironment near him and actually changed the immune system to bring cells that are not attacking him, actually quite supporting him. He's doing two tricks. First of all, he presenting protein and said, hey, I'm self. I'm one of the cells from the body, don't attack me. Second, he produced specific proteins that exhausting the immune cells to attacking him. Make the immune cell weaker, okay? How cannabis affect that? If cannabis reducing, so we're giving a treatment that actually activating the immune system. But cannabis reduced the immune system activity in inflammatory bowel disease, in autoimmune disease, I showed you in the leukemia. So if you look on the literature, okay, if you look on the literature, there are few publications that show that patients, these are clinical trials, patients that getting immunotherapy and cannabis, it's actually improve the activity of the immune therapy. This is beautiful. But if you look, there are other papers that showing that if you, again, clinical trials, that if you give cannabis together with immunotherapy, it's reduced the activity of the immunotherapy. My problem with these publications that the same idiot published all of them. Okay? If you sitting far, of course, I wouldn't say idiot if it wasn't me, okay? We did 4 clinical trials with different results, but this is fit to what I believe. cannabis is not one plant. There is many varieties, and part of the varieties can do something, and part of the varieties can do something else. So we ask ourselves, okay, let's build a model for that. Okay, let's take mice, normal mice, and let's take mice without immune system. Every scientist knows that we are using mice with no immune system. We call them nude mice because they don't have a hair at all, okay? So we're taking normal mice and nude mice, and we're treating them with cannabis. We create them tumors in these mice, and we're treating the normal mice with cannabis, and we're looking for strains that reducing the size of the tumor. The same one that succeed here, we're treating with mice with no immune system, and now we want to see which extract reduce the size in normal mice, but don't affecting the size of the tumor in mice without immune system. And that will tell us that it happened through the immune system. We succeed to isolate specific extract. We learn the mechanism of action, what it's doing. We succeed to isolate specific molecules that doing that effect. We define that it's reducing secretion for the cancer cell of specific cytokines. I don't want to go deeper to the results, but we saw that these molecules affecting the immune activity of the cells. We take it and we mimic it, what we did with the full extract, and we see that the single molecule doing the same effect as the whole extract. Then we know that what the cancer cell, and I explained that a few minutes ago, what the cancer cell doing, is doing two things. First of all, he presenting protein cell, I'm self, this is me, don't attack me. Second, he produce specific protein that affecting the immune cells to be weaker. Actually, this protein breaking down an amino acid that is essential for the T cells to attack him. We have very good treatments today, what we call immunotherapy, is that blocking anti-PD-L1, Keytruda, whatever you call them, that blocking the ability of the cell to say, I'm self, on this one. What we find out that the cannabis blocking this part. So now, if you're giving cannabis, specific cannabis, with this specific molecule, together with immunotherapy, you get a way better action than the immunotherapy alone on the cannabis alone. But what is the message here? We need to know. It's not enough to say to a cancer patient that getting immunotherapy, take cannabis. Which type of cannabis? What molecules? What is the effect? It can be on that side or it can be on that side. Cannabis is a complicated plant, but all the plants are complicated, okay? I used to say in one of my talk previous, like 7 years ago, cannabis is not like a tobacco that have nicotine. There is a lot of molecules. Then I met a guy from Philip Morris that told me, oh, you know, cannabis is a simple. In tobacco, there is more than 1,500 active compounds. So I stopped saying that anymore, okay? All the plants are complicated. When we are going to the grocery store and buy apples, we know that the green one called Smith, they are very sour. The red one called Macintosh, they are sweeter, and so on and so on. We know that apples are different. Israelis have something very unique to us. We have superpower. When we knock on watermelon, we know if it's sweet or no, okay? Every Israeli, when you're buying a watermelon, if you travel in Israel, you see this phenomena. I'm serious. We're knocking on the watermelon to know if it's sweet or not, right? I have like 3 Israelis here that can approve me. So every Israeli know that every watermelon is different. But when we're going to herbal medicine, we flatten that. It doesn't matter if it's cannabis. It can be archinicea, valeriana, ginkgo, whatever. Archinicea good for the immune system. What do you mean? Ginkgo good for that. What do you mean? Like all of them the same? Doesn't matter where it's growing. We have the same molecules. Guys, it's the same story as cannabis. When we're talking about today on psilocybin mushroom or magic mushrooms, all of them are the same. There are more than 20 families, hundreds of species, many active compounds called tryptamine, the same story. And this leading me to what we called the entourage effect. Entourage effect is mainly saying in cannabis or other plants, there are many compounds that are doing something complicated. But I think in cannabis, it's unique. There are 3 layers of the entourage effect. So first of all, let's start, what is the entourage effect? Ravi Meshulam and Professor Ben Shabbat published in 1998 a paper. Lumer, I'm not sure if you were there or not, no. So published a paper called An Entourage Effect. After these 3 words, try to read it. It will be difficult to all of you. Okay, inactive endogenous fatty acid glycerol ester enhanced to arachidonic glycerol cannabinoid activity. None of you remember that. You remember the entourage effect. Actually, it wasn't about cannabis. It was about endocannabinoids. And he showed the 2 endocannabinoids, 2 linol glycine or 2 palmitotic glycine, which not binding CB1, enhancing other endocannabinoids to AG to bind the receptor. And it called it the entourage effect. It wasn't about cannabis. I think, Ethan, you were the one that took it, no? Okay. So Ethan here is saying that the next year they sent another paper that saying that it's also applying for the plant. But the first one was this one, okay? So what is the entourage effect? So first of all, I showed you, for examples in the leukemia, that you need 3 molecules. What the meaning of these 3 molecules? When 3 molecules activate on the same cell, different receptors, we actually call it polypharmacology. It's when you're treating a disease by modulation, one target, you have activity which is more complicated. I showed that in the leukemia example, how 3 molecules binding different receptors, opening calcium channels. I didn't show the mechanism of action, but they're doing something synergistic in the plant, in the cell, but they're affecting the same plant, the same cell. And if you think about that, plants are harboring polypharmacology within. Because if you think about a physician 10,000 years ago, 5,000 years ago, if you come with a kind of inflammation, you will take a root from here, a flower from here, a stem from here, combine them together and kind of creating polypharmacology. And I think polypharmacology will grow and be bigger and bigger in treatment, and also I think the FDA will start to approve more and more. This is my opinion, because what's changing in the last 20 years, our analytical ability to analyze these molecules, but more than that, to use computers to understand networking and pathways. So if you look about network of 3 molecules, 5 molecules, if in the past it was complicated to understand that, today with computer analysis, it's quite to understand and to see what is the drug-drug interaction and modeling and things that will open that gate. But this is, for me, the first layer of the entourage effect. Few molecules that affecting one cell. Second layer of the entourage effect is that we have molecules that affecting different cells. So I will take, for example, IBD, inflammatory bowel disease. Inflammatory bowel disease consists mainly 2 types of cells. It's the colon cells that being somehow harmed, mainly probably by bacteria or microenvironment, whatever, but they're being harmed. They're secreting specific molecules, so-called cytokines, calling other types of cells, the immune cells, to come and to help them, but actually we're creating chronic inflammation that cause this disease, okay? But it's 2 types of cells, 2 systems, the colon cells and the immune cells. So in 2 different studies, 2 different PhD students in my lab, one of them work on the colon cells and define specific combination of cannabinoids, actually 2, that reduce the ability of the colon cells to secrete these cytokines to call the immune cells. But in the same extract, different student, different PhD work on the immune cells and show that different molecules from the same extract actually blocking the ability of the immune cells to migrate towards the colon. Different compound, different results, okay? The first one, if if people are here are physician or scientist. First one, reducing specific protein called STAT3, which is creating the inflammation or the release of cytokines. The second one, reducing protein called CXCR4, which actually it's kind of the antenna. The colon cells secreting the molecules that reaching the immune cells, binding them, and they're binding a specific protein called CXCR4, which feel now where it's bind, if it's bind here, here, and here, and started to migrate towards. So the cannabis, from one hand, one molecule from this extract reducing the colon cells to secrete these molecules, different molecules, blocking the ability of the cells to feel it and to migrate, and you're getting entourage effect, you're getting synergism, you're getting powerful reaction that if you take just with molecules that are affecting the colon, or just these molecules that are affecting the immune system, you won't get it. And this is a different layer, more complicated for me, for the FDA or something like that. It will be two treatments that we need to combine or we need to use the plant. But this is a different layer of the entourage effect. The third layer of the entourage effect, or third entourage, it's when we are not talking about single indication, but we are talking about the quality of life of the patient. We are talking about the whole patient. So if we take pain patient, we did a clinical trial of 3,800 pain patient, and we measure many parameters of the patient. Even though the patient, 50% of the patients said the cannabis has really helped them change their life, did a lot. When we measured the pain, we saw that it didn't reduce much of the pain. Less than 30% reduced 2 points in the pain of the patient. But when you started to check why this patient said that it's really helped them, we saw that it's really also reduced anxiety, reduced depression, improved sleep, improved appetite. And when you measure quality of life action, what we call catastrophism, how the patient conceived his disease, then we saw a huge improvement. Actually, the cannabis improved the quality of life of the patient. But if you think about that, few molecules affect the pain, few molecules affect appetite, few molecules affect anxiety, few molecules affect sleep, and altogether, the patient improves his quality of life. And this is cannabis because it's affecting so many systems. In all of them, is doing moderate action, a little bit on the pain, a little bit on the sleep, a little bit on the appetite. You're getting something as a whole treatment, which is better, and you wouldn't get it from single molecule, single treatment. You need to give pill for sleep, pill for pain, pill for appetite, and so on. Cannabis gives something more holistic. So if we think about that, cannabis is kind of an orchestra of molecules that working on a system called the Endocannabinoid System, mimicking things in our body. We can narrow it down. So I didn't prepare my talk until 2 hours ago. And I prepared because things that I heard during this conference, there are kind of division here. People that coming from pharma and looking for, let's take single molecules or there and purify it and go with that. And people that believe more in the plant and looking about the entourage effect. But actually what I'm trying to show that nobody is wrong here. In few indications, if you're looking very specific, you want to treat something very specific, you can isolate a molecule and go pharma route, and there is indication that it might work. If you want to be more complicated, more risky on the business side, you can say, okay, I will take these two molecules that are affecting the colon, these two molecules that are affecting the immune cells, I will try to combine them to botanic treatment or to be very, very brave and to try to go with this to the FDA. Congratulations to you, okay? But it's still, I believe in that, okay? And the third one is to say, when you want to treat quality of life of the patient, which is it's the majority of our patient, you can't run away from the plant. You might want a consistently a product, something safe, something that you know what you get, but you won't get away from the plant because it's too complicated and it's doing too many things. I think the future will consist part of these things. I think we will see more and more drugs, FDA approved, single molecules, 3315, that Dedi wrote a patent and somebody licensed it and go with that to reduce estrogen, okay? We will see drugs like that. We will see the whole plant as medicines for many years. I don't think we can run away from that. It's already there. Countries like Israel, 150,000 prescription a month for patients, one of the most prescribed medicine in Israel. But this is the whole plant. And I do believe that we will see a new route, a new path of more complicated drugs that trying to affect not one mechanism, not one system, but more of them. And I think it's, you need to break a wall, you need to be innovative, you need to do that. But I do believe that there are diseases like ALS, Alzheimer, types of cancer that we hit our head to the wall many years and don't succeed to solve them with a simplified activity. And when we will understand the complexity and we try to do complicated things, we might bring a cure to these ones. By that, I want to say thanks a lot to the amazing team that's working. We shrinked, that was before the corona, we shrinked a lot from then, but these are my students and my workers that are doing the amazing work that I showed. Thank you very much.
Thank you very much, Dedi, for this wonderful presentation. But I have to say that you left me a little bit confused. What do you think? What does the future hold for cannabis? Do you see cannabis being developed to be a pharmaceutical drug, or rather it might just, or not just, but it might stay as a herbal extract or product? What do you think?
So I think it depends on the indication. In my opinion, and my father taught me that if I'm arguing with somebody, I'm 50% wrong, okay? Actually, he would say, and if you argue with your mother, it's 100% wrong, so don't argue. That was what he used to say. So it's my opinion, and just my opinion. I believe that in specific indication, and mainly save lives indication, it will be more precise, more pharma route, these molecules for leukemia or whatever. I think in indication which is more related to quality of life, which more wide, will stay with the plant for two reasons. First of all, it's a cheap medicine. And there is a reason why, in the global world, I just came from India yesterday. I actually fly from India here. I learned that Miami is not on the way from India, right? Or Israel, it's actually between. There are countries like India that cheap medicine is extremely important. I think cannabis is a cheap medicine and it stay like that. And I think in the complicated thing, in the, quality of life, things that you're treating it will stay with the plant. I think we need to precise it. I need to know which extract or which plant to use to what, but I think it will stay with the plant.
Thank you. Thank you. I would just like to comment that there might be some difficulties in this approach, since once you define a certain substance, a certain molecule as a drug, then it comes to be under regulation, which might not support public use for different purposes. So just,
So again, the cannabis is very unique because there is regulation. Each country looks in a different way. But I just came from India that they have regulation for plants. They have plants as a medicine, okay? I forgot how they call it, Avida or Avida, you know, Ethan? Ayurveda, okay? It's a specific regulation for plant, which we don't have in the States or in Israel. So it's very depend on the regulation. I agree with you. There is problems. Bonnie.
Hi, Dedi, thank you. That overview is so important. A question about the so-called minor cannabinoids that you've been testing. Are they much more potent than the major cannabinoids? Do you need to amplify their concentration or dose that you're giving in these models, or do you give them in relatively tiny doses like they occur in the plant?
So the answer is specific for each minor cannabinoids that we define, okay? There are minor cannabinoids that we found which are extremely potent, and more like 3318 in the leukemia is very similar to CBD. When you increase its amount, it's more potent to from the CBD. So even if you tried it on epilepsy, it's working even better than the CBD, okay? Very similar action to the CBD, but something else there. There are other phytocannabinoids that I think their potency is lower, but they are potent. Like the minor, there are many minor cannabinoids. The minor cannabinoids that we found that active, they were very potent. The other one that I showed, the estrogen, because you have three oxygens on the outside, its bioavailability is way higher than a regular phytocannabinoid, and that might be why it's more potent. I don't know the result, but yeah, they are potent.
Thank you.
Bonnie,
Hi,
thanks.
Dedi. Thank you for a great lecture as usual. I have a question going back to the slide where you showed the cancer cell having two actions, one saying, I'm self, don't kill me, and that you're saying that that's when the anti-PD-1 comes in,
Right.
that is not, is this new information that you're sharing that with, you said a new paper, that's not where cannabis is causing an issue, it's causing an issue more on the immune system in the microenvironment?
Yeah, the cannabis is what we found, okay? Not the cannabis, this cannabinoid is affecting the cancer cell. First of all, is reducing the secretion of specific cytokine called CSF1. And CSF1 changing, the cancer cell secretes CSF1 to change the profile of the macrophages that are arriving the microenvironment. M2 instead of M1, so actually there are macrophages that coming and deactivating the T cells, changing the profile of that, changing the activity of the T cells. That's one thing. The second, it's reducing the activity of two enzymes, inose and arginase, which break down arginine, which that's the way to do what we call immunosuppressive. Blocking the ability of the cancer cell to immunosuppress the T cells activity. And so if we take tumors, and we did that, you take tumors from patient, you grow them in a plate, you treat them with the cannabis, okay? And then you're taking the supernatant, what they secreted and put on T cells. You see that if you're not doing that, it's inhibiting the T cells activity. But if you treat with cannabis, it won't inhibit the T cell activity. This is like classic method to test that.
Okay, so I have a follow-up question, and it's okay if you don't want to answer me. Just today, I got a text message from the mother of a boy I've already emailed you about once. I don't know if you remember. It's a 10-year-old with metastatic osteosarcoma. His second cancer, he's treated for leukemia as a young child and has a subsequent secondary cancer. He was on cannabis and chemotherapy. His physicians decided to switch him to nivolumab, and I discontinued the cannabis, and she texted me today to say, is there any way we can add it back? He has the most horrific side effects. And so I wrote back, I don't know if there's anything safe, but I'll see Dr. Meiri and ask him. So I know I'm putting you on the spot to translate to a cancer patient.
So it's always difficult for me to answer that. The clinical trial that we showed that was reduced the activity of the immune cell, the immunotherapy was on that drug, specific drug. And we saw that in general, the success rate reduced from 40 few percent to 20 few percent. So it's really affecting that. But this was high THC strain classic. The strain that we used here in this study is high CBG strain. So it's totally different profile, totally different. In Israel, I'm working with all the head of the oncology department in the main hospitals. And we had a panel of all the head of the oncologists and me about that. And the answer was, there is risk, we won't take it. So in the frame that they gave immunotherapy, that's stopping the cannabis. I do think, according to our results, that the high CBG strain today that are available outside are safe enough, and I don't see this reaction from the patient and from the preclinicals that we get. But I'm not sure that these strains are effective on the side effects. So I don't know if the high CBG strain are really reducing nausea or pain or that. I don't know their effect on the side effects. So it's complicated. But there is risk to give cannabis with immunotherapy if it's just cannabis.
Right, but again, it depends on the cannabinoids. So,
right.
and that's a nice, helpful answer, because if he's terribly desperate and he's not finding any relief, I would probably feel a little more comfortable. We can try CBG.
So again, the cannabinoids that I showed in the immunotherapy work was CBG. It's not a minor cannabinoid. I don't call my CBG minor. So.
Okay, thank you very much.
Just a comment, Bonnie. It's such a tough question, but then you have to weigh the other side. Is that, can this kid complete the course of therapy without the help of cannabis, or are they going to have to throw in the towel on immunotherapy?
Thank you, sir. I just wanted to comment on a couple of things from your previous slide regarding the approach moving forward as far as investigating this further. Groupers versus splitters. Those that tend to want to treat it holistically versus those that want to pinpoint specific parts of the plant. I wonder if there's a middle ground regarding this. And considering that there are different strains, just like your apple slide, could it not be more a way of approaching it in understanding that our categories, neuromuscular, neurological, GI, pain, inflammation, as specific areas to address, rather than getting specific conditions or illnesses, rather grouping them in certain generalities that we can pinpoint and direct treatment plans for patients? Do you feel that rather than, again, segregating the plant and cutting it up in smaller pieces, why not just develop certain specific medical strains towards that grouping and see and investigate that very question of entourage and how mental health affects all that, and how the mental health, if improved, can improve the physical condition?
So I will answer that in two ways. First of all, it's, sometimes the truth is not the nice there. I'm a cell biologist, that's what I'm doing. So I'm interested in what the molecule doing in the cell in the end. That's who I am, okay? I'm not the cannabis, regulator or something like that. But to answer that, the technology is there. Like, we can analyze the compounds, we can say this is the profile, this is the groups that doing for that and that. The technology is for there, but the knowledge is not there yet. So we don't have the knowledge to say in all the indication, not even in minor indications, to say this is the profile that you need for that, or this is the profile that you to group that. We just don't have the knowledge yet. So, I showed few things that we have results, but in most of the things we don't know. And, I'm responsible for courses in Israel for physicians or for nurses, and people said, yeah, education is very important, we need to teach them. What I know to teach them, I don't know what to teach them. What I'm showing the Entourage, the Endocannabinoid System there, but I don't know to tell them what to prescribe, which one is really good for that or for that. I'm starting to have a knowledge. So we don't have this profile at. The technology is there. I must say that very few scientists in the world, I'm not talking about companies or that, I'm talking about even scientists, taking the approach to really go deep and to look on the minor cannabinoids and the combination. You know, Heinanit was sitting here and other scientists doing that, but these are, I can count them on one hand. So we don't have this data, we just don't have this data, okay? And that's reality. We don't have the data to push it there. Thank you very much and have a good